What's the greatest work of poetry ever written in the English language? You can tell I'm a college professor because this is a multiple choice question. There's only one answer. What's the greatest poem ever written?
Beowulf.
Excellent choice. Wasn't written in English originally, though. I've got it here. It was in Old English, that's right. I've got the correct answer here, though. It's Milton's Paradise Lost. The other possibility is Spenser's Faerie Queene. But Milton's Paradise Lost. This is, it's one of my loveliest possessions, I suppose. It's a miniature edition of Paradise Lost. And this is the edition that a young naturalist carried with him when he circumnavigated the globe in the 1830s. So Darwin apparently had a field jacket cut for the voyage on the Beagle. And he had a special pocket sewn into it into which he could slip these miniature editions. And he loved Paradise Lost. And he committed large chunks of the poem to memory. And there's a vein of scholarship that actually suggests that some of the patterns in Paradise Lost are actually replicated in Darwin's On the Origin of Species, published in 1859. It is a miniature edition, and Darwin's eyes were much better then than mine are now, so I'll have to take my glasses off, and I'm just going to read you a few lines from the poem. Need a microscope for this part of the talk, actually. In this pleasant soil, his far more pleasant garden God ordained. Out of the fertile ground he caused to grow all trees of noblest kind for sight, smell, taste, and all amid them stood the tree of life, high eminent, blooming ambrosial fruit of vegetable gold, and next to life, our death, the tree of knowledge grew fast by. I love this particular line. And all amid them stood the tree of life. And that's really a metaphor that I use throughout the book, the amoeba in the room. If we think about Eden, it's easy, I think, to revel in the idea of an Eden when we're in a tropical rainforest or when we're where? Snorkeling over a coral reef. Less so when we're just going about our daily business in a city. But yet this theme, and all amid them stood the tree of life, is actually true. It's true of us right now, and it's true of you as you sit and me as I stand. So rather than talking about a tropical rainforest, I'm going to introduce to you a little garden pond. It's my garden pond in Ohio. I live close to Cincinnati. And a PowerPoint would be useless here. You can picture just a little pond. 6 fish swim around the ripples from a little pump that circulates the water. Frogs visit from time to time and are attacked by my cats. This doesn't seem like a place of teeming biological diversity, but when we look more closely, it becomes clear that the little pond is a place of quite mind-blowing biological riches. The water contains trillions of bacteria, 10 times more viruses, and the diversity of eukaryotes is incredible. Eukaryotes being organisms like us, our cells. We have 23 pairs of chromosomes that are packaged in a nucleus in each of our cells. And other eukaryotes then also have a nucleus as the house where their chromosomes are stuffed. And I'll talk a little bit about these unicellular or microbial eukaryotes shortly. But looked at from a broad perspective, we discover that every kind of living thing really is represented in this pond. And all amid them stood the tree of life. We don't have to go to a rainforest. There are, for example, in the pond, multiple amoebozoans, cryptomonad algae, diatoms and water molds, dinoflagellates, gorgeous shelled amoebae called rhizarians, colonies of sparkling green algae, and an apple green alga called a euglenoid that glides through the water. And if you look at these organisms, if you take a little bit of time, as I have done, to look at them under the microscope, they are absolutely beautiful. This is one of life's great experiences. At this time of year, before the leaves are out in my part of the country, my pond turns this bright apple green, and it's because of the billions and billions of these euglenoid algae that bloom under these conditions of quite high light intensity. And looked at under the microscope, each one of these cells is like a Valentine's box of candy. They're heart-shaped, and they move, they sway to and fro, and as they swim, they rotate, and then they also swim through this wider gyre. They're absolutely beautiful. The dinoflagellates in the pond have got little eyes called eye spots, and they use these structures to find the optimal position in the water column for their photosynthetic activity. There is a dinoflagellate. Unfortunately, it doesn't grow in my pond, but you could find it in the ocean here in California. In fact, that's where it's been studied. It's a particular dinoflagellate. It's a single cell. It's a single eukaryote cell, but it has an eye sticking out from one side. It's a modified eye spot, and absolutely beautiful cell. And it really is an eye. We don't really know what it does, but it is an eye. It has a crystalline lens. It's got a photoreactive cup, and some beautiful work at Scripps Oceanographic Institute in the 1960s was done by this sort of micro-ophthalmologist, where the scientist actually flicked out these eyes from these individual cells and did some, probably is it this one or this one? He figured out that it had a 30-degree field of view, and that this single cell could actually perceive oncoming predators at a distance of a few centimeters, so something like a copepod, rather than a sperm whale, I suppose, and could swim out of the way of the predator. It's a single cell, and it's got this structure that looks like an eye and is an eye, although we don't really know everything about what it does. In addition to the fish and the frogs in my pond, there are microscopic animals, including nematode worms, water bears, and rotifers. And when biologists carve up organisms into big slices referred to as supergroups, so these are much larger groups than kingdoms of organisms, we find, in fact, that the animals are united with the fungi in a single supergroup called the opisthokonts. The animal relatives in my pond, then, these fungi actually swim around with undocumented abundance. We know very, very little about the biology of these organisms, and these are fungi that have cilia. They're rather like human sperm cells, and they swim around in the water. There's far more genetic diversity in the pond in the form of bacteria and archaea that are like bacteria in that they lack nuclei. This is a separate supergrouping of organisms. And then that genetic diversity, again, is eclipsed by the viruses, because for every cell in the pond, there are at least 10 to 100 viruses that prey upon that cell. Tremendous genetic diversity in the pond. And here's something that's surprising. After centuries of taxonomic inquiry and using the latest molecular genetic methods, so these are referred to as metagenomic methods, we're still unable to document all of the life in my pond, which is why it should gain national park status soon. But we could say the same, probably, about, I'm sure we could find a pond around here pretty quickly. And all amid them stood the tree of life. It's interesting with these metagenomic methods that I write about these methods in the book, trying to boil things down to some sort of straightforward descriptions. The cost of these methods has plunged. It's absolutely an astonishing technological achievement, business achievement too. About 10 years ago, it cost $95 million to sequence a human-sized genome. Today, we can sequence, I can do this, scientists can sequence a human-sized genome for less than $6,000. So I thought about this a bit, and I thought, if we could apply the same economies to the automobile industry, I could go out and purchase a brand new Rolls-Royce for less than the cost of a Thanksgiving turkey. But unfortunately, I don't drive a Rolls-Royce. Hasn't happened yet. So we'll all be having our genome sequenced pretty soon. But even with this technology, the genetic variation, the abundance of organisms in the pond, over 1 trillion cells, it's impossible. We could keep sequencing, and I don't think we'd ever get to the bottom of it. Earth, air, and sea. Let me just talk a little bit about the spectacular numbers and diversity of microorganisms in different locations. Each pinch of soil teems with tens of millions of bacteria and hundreds of millions of viruses, and is permeated with networks of fungal filaments. These organisms control soil fertility and support the growth of plants, while other microorganisms coat the surface of plants and also colonize their internal tissues. So when we look at an individual plant, a rose, in fact, we're looking at a rich biological community. We're not just looking at one organism. And the same is true of us. Trillions of bacteria, as I'm sure you're aware, trillions of bacteria live in our guts, and they affect every aspect of our well-being. And I'll come back to the human microbiome in a couple of minutes. Our air supply is microbial too. Spores of fungi float into our lungs with every breath, and there are plenty of them to go round, with an estimated 50 million tons of spores ejected into the atmosphere every year. And that's not just a guess. There are reasons why we could talk about this. It's beautiful science that actually allows us to come up with this admittedly ballpark estimate. But 50 million tons of spores, 50 megatons of spores flung into the atmosphere every year. These particles are the bane of the asthmatic's existence. They affect the chemistry of the air, and they even influence patterns of rainfall. I've got a graduate student, doctoral student in Ohio that's actually looking at this, some of the chemistry involved in that extraordinary claim that microorganisms can affect rainfall patterns. Seawater. Seawater is a soup of microbes. Most of us love documentaries about killer whales. Sharks are, of course, always a reliable draw on television. And it's easy to ignore the fact that microbes run the show for the animals. A cup of seawater contains 100 million bacteria, billions of predatory viruses. There's a single kind of photosynthetic bacterium that absorbs more carbon dioxide than all of the world's rainforests. A single kind of cyanobacterium. It grows, flourishes in the open ocean. It's a greater refrigerant than all of the world's tropical rainforests. Diatoms, actually, marine diatoms, those beautiful cells, these circular centric diatoms, they form these glass boxes, beautiful things to look at under the microscope. But they're responsible for about the same amount of photosynthesis as these cyanobacteria. So together, if we look at the diatoms and the cyanobacteria, they're doing more to control the climate than all of the green plants on land. Microbes get more airplay in extreme environments where nothing else lives. There are these archaea that I mentioned earlier. They're a separate grouping of organisms from the bacteria. This was recognized, very controversial at the time, it was recognized by a scientist called Carl Woese in the 1970s, that the archaea were a separate group of organisms that lacked nuclei. They're prolific. They're almost everywhere too, but we tend to associate them with extreme environments. So there's an archaea that lives, is capable of cell division at 121 degrees Celsius, which is interesting because 121 degrees is the temperature that we use in a lab or hospital autoclave to sterilize instruments. So this organism, it's called Pyrolobus. Actually, it's a strain called strain 121 because it divides at 121 degrees C. And in the paper that first described this, it was great witticism for a peer-reviewed scientific paper. It said that it's chilled below 90 degrees because it quits dividing. So it has to work between 90 and 121 degrees. Cold environments. There's another archaea that divides only once a month at the bottom of an Antarctic lake. Methanogenium, I think it's frigidum is its Latin name. the most acid-tolerant thing. There's an archaea that can grow at pH 0.06. That's more acidic than battery acid. How is this possible? Radioactive resistance. There's a bacterium called Dinococcus that survives 15,000 grays of ionizing radiation. Humans are toast at 5 grays, total absorbance. So this thing can survive. And actually, the reason we know that is tragic, but interesting. But it's from studies of the very careful studies of the first responders to the Chernobyl disaster in the 1980s. We know exactly what kind of dose. We know exactly what dose of radioactivity they received, and we can look at the consequences to their health. We don't have to go to such extreme places to find extreme or interesting environments. Our own homes, our bathrooms, and our laundry rooms are places where we find incredible microbial diversity. We think about a bathroom and a hot water faucet. I mean, there are lots and lots of microorganisms, including fungi. There are very hot places then in a bathroom. There are very alkaline conditions that we find in a laundry room, laundry detergents. Very, very alkaline conditions, and you'll find incredible diversity of microbes in these places. A dishwasher is a place of extraordinary diversity and yet to be accorded national park status. These organisms make extraterrestrial life seem plausible. And the current interest is in Jupiter's moon Europa, Saturn's moon Titan, and then all of these Goldilocks planets that are discovered every week we hear of new Goldilocks planets. So, how have we learned all this? Microbes are everywhere, but invisible, or they were so before the invention of the microscope in the 17th century. The revelation that there was more to life than the immediately observable was a discovery of immense significance. And yet this was sidelined by a continuing, narcissism, self-obsession with our own specialness. And Linnaeus and other natural historians of the 18th century catalogued plants and animals and just ignored the microscopic majority. Linnaeus was particularly upset with the fungi. He had no interest in them. What did he call them? I think he gave them a name at one edition of the Systema Naturae. In the 18th century, he said, we'll just call them all one thing, fungus, I do forget. Chaos fungorum, all of the fungi. Chaos fungorum, we're just going to ignore them. When microbiological inquiry was re-stimulated by Louis Pasteur in the 19th century, it became the science of germs and infectious disease. The point was not to glory in the diversity of microbes, but to exterminate them. And so, even in the 19th century, most of life was disregarded. And we still teach biology. Most of us, most biology teachers, K through 12 and in college too, teach biology from a very macrobiological perspective that elevates humanity and sort of situates us as the triumph of a 3.5 billion year old evolutionary process. It's interesting if you look at, you can Google image this or go into a school classroom and look at trees of life. And it's amazing, and still in many, many textbooks too that you've got, it's mostly animals, and most of the real genetic diversity and metabolic diversity of life is hidden under an amoeba or a hairy bacterium at the base of the tree. This is still the way that we look at biology. Pliny the Elder, was it the Elder? Yeah, Pliny the Elder died in AD 79, Vesuvius. He was so interested in seeing the volcano, anyway, got too close and he perished. But Pliny the Elder published the Historia Naturalis, and you can buy this in the Loeb Classics edition. It's 12 volumes, I think. And in this book, Pliny had this very modest goal of documenting everything in the world. And he got to 12, 13 volumes, and he perished at Vesuvius. He didn't quite get there. And it's actually a fascinating read, or parts of it are. He describes all these strange animals that are based on descriptions from travelers at the time, many-headed monsters and snakes that could destroy you by looking at you or make you pregnant, or amazing things. Fascinating book. So it's obviously futile, Pliny-esque to engage in this idea of just documenting everything, thinking that we could know everything. But the thing is, I think we're still doing this. And if I give this talk in Boston, I have to be very careful what I say, because the deity that is E.O. Wilson is the Harvard professor there. But E.O. Wilson put his name behind it, and a brilliant biologist, ant specialist. E.O. Wilson put his name behind the tree of the Encyclopedia of Life project some years ago. It's really, really interesting, and this exists online. And scientists can upload descriptions of different organisms. And in the original sort of mission statement, it reads just like Pliny. We're going to describe everything. We're going to put a lot of resources into this, and we're going to figure out what's on Earth. Figure out all the species, name them all. And if you go on there, what you find is that most of the time it's new species of crabs and lobsters, big things that have been described. There's a few microorganisms there, but it's mostly animals. There are some really big names in biology in Europe too. Lord May of the Royal Society, he had a paper in Science, I think it was last year, but there have been a number of these sort of calls to action where we need to, and the argument is that we need to document all of the species on Earth, and that we need to put what would be hundreds of millions of dollars into this activity, global activity, to name everything. There are so many problems with that enterprise that I'm only going to tell you about two of them. One of the problems is that it's impossible. Among the microorganisms, for most of life, we really don't know what a species is. There's no species concept. It's philosophically unsound. We can't possibly document species when we really don't have a proper definition of what a species is. Most of life doesn't operate like us. So that's the first thing. It's futile on those grounds. The other thing is, why would we do this? Why would we take funding from other areas and engage in this Pliny-esque cataloging of biological diversity? What, before it all goes extinct? What's the rationale for doing this? And actually, one of the papers actually says that it's only by knowing how many species are out there that we can properly document rates of extinction. This seems to me to be perhaps even morally reprehensible. We're trying to create sort of some stone tablets or e-tablets that the aliens can come and find them some years from now and say, boy, look at what a mess they made. They managed to kill 10 million species in a short space of time. Anyway, medicine, more positive news. Each of us carries a microbiome of 100 trillion, actually probably 1 quadrillion is the number that's going to be published, going to be discussed more frequently in the future, about 1 quadrillion bacteria in our guts. Kilogram or so of pure bacteria in us right now. Along with a smaller number of archaea. There are archaea in our guts that are responsible for methane production, and lots of single-celled eukaryotes too, including fungi, and we don't know what most of them do. What do the known knowns do? So when we eat plant carbohydrates, complicated sugar-based molecules, the human genome actually lacks the instructions for breaking down those materials. We're very, very good. We're actually better than the other great apes, surviving great apes, at breaking down starch. But when it comes to the other large polysaccharides, we can't break them down. We don't produce the enzymes necessary to break those materials down. Those materials are actually, the enzymes to break them down are released by bacteria in the gut, and These bacteria then release short-chain fatty acids, little molecules that fuel our metabolism. We can burn those things. Errant populations of bacteria are linked to all kinds of autoimmune diseases. There may be a relationship between asthma and disturbance to the infant microbiome. So these organisms are important for our digestive activities, but also for our health and well-being. More surprising still are the results of experiments on mice which suggest that aspects of our behavior, particularly levels of anxiety, may be modulated by organisms in the gut. These are absolutely beautiful experiments where there are these facilities, they're notobiotic lines of mice, rats also, that lack bacteria in the guts, and they have to be given a special diet so that they can absorb enough calories to keep going. But what they've found in these experiments, or with observing these animals, is the ones without the bacteria in their guts are less anxious. They do all kinds of weird things that mice wouldn't normally do. So if you put them in a maze, they say, oh, okay, they do. They say to one another, we're just going to wander around here, but we're not going to get stressed out. And they don't seem to be worried by dark places, and they're just not doing a lot of twitching of their whiskers. And so there you go. These bacteria in the mouse gut seem to modulate the behavior of the mice, and what goes for mice surely goes for man, or likely. So when you eat a slice of toast in the morning, full of complex plant carbohydrates in the wheat, and perhaps if you look out of the window and you feel happy, who's sending the signals? Because the calories in that toast are not going to be released, so you're going to get a sugar burst from the starch, but most of the calories in that toast are not going to be released for hours after you swallow it. Last point, philosophy. Part of my job is administrative, that I run this undergraduate program, and it's the Western program, and it's an interdisciplinary program. It exposes me to lots and lots of different subject areas. Our students study, certainly some of them study scientific subjects, but others are studying all kinds of other things. And so I have to become quite adept at bluffing my way through different meetings with faculty so that they don't know that I have no clue about their own areas of specialization, that I'm a scientist. And so I have to come up with different phrases that I can use that seem to suggest that I'm learned in their areas. And so for anybody that's a theologist, I always use the word ecumenical, because nobody knows what it means. I don't know what it means. For philosophers, it's Cartesian. Yeah, that would be Cartesian, wouldn't it? That's a Cartesian way of looking. You can apply that to changing the oil in your car, I think. It's great. That should be the next book. I'll come up with, phrases for bluffing your way out of faculty meetings. But anyway, speaking of philosophy, vast knowledge in this area, we are, as I've described then in the last few minutes, mobile ecosystems. In the 17th century, Descartes said, I think, therefore I am. But this faith in the integrity of the individual seems a bit naive in light of all that the new biology is teaching us. So I'm just going to read a brief excerpt here and then open this to questions. Surveying the solar system for its natural resources, the perceptive extraterrestrial would report that our planet is swarming with viral and bacterial genes. The visitor might comment that a few of these genes have been strung together into large assemblies capable of running around and branching toward the sunlight. It's time, I believe, for us to embrace this kind of objectivity and recognize that the macrobiological bias that drives our exploration and teaching of biology is no more sensible than trying to evaluate English literature by reading nothing but a Harry Potter book. The science of biology would benefit from a philosophical reboot. And I do thank you for taking time out of your busy schedules to come and listen to me talk about microbes for half an hour. So thank you.